Yeah, so good afternoon, everybody. It's really, truly an honor to be, a participant in this amazing session. We've just wanted to take a little bit of time to recognize, all the different impacts that Eric Graham has had, on our lives and our careers. And as Suzette said, my name is Jessica Blois. I'm a associate professor and vertebrate paleontologist at University of California, Merced. I turn it over to Simon.
And I'm Simon Goring, and I am an associate scientist at the University of Wisconsin. And I guess we're going to talk a lot about, I don't have the speaker notes, Jessica is on her screen. And so Jessica, I'm sorry, but I can't remember who was supposed to say this, but we're going to talk a lot about the influence that Eric had on both of us. But I think one of the sort of funnier things is that as we were coming up with the title for this talk, you'll see that we have like Neotoma administrators as if it were a Latin thing. And we actually went and searched the proper Latin form of the plural for administrators, just because we knew that Eric was such a stickler for taxonomy. I think if anyone has ever heard Eric's discussions about pollen taxonomy and how they reflect systematics much better than taxonomists, I'm sure you'll appreciate that. So
awesome. Yeah, so we'll try to keep it short. We know we're the last talk. It's been a long period, but we're gonna briefly go over our history and roles with Neotoma, some of the brief lessons we've learned, which a lot of them have already been covered here by Jack, by Andrea, by many others. And then a little bit of what we see is, and which I think echoes, again, what everybody else is saying, just sort of the legacy of Eric, scaling from local to global, with both data and with people. Okay, so our introduction to Neotoma. I met Eric in 2009 when I started a postdoc working with Jack Williams. And this is a grant mentioned, I think, by Russ. Russ is the person who actually pointed me onto this grant. They had an NSF postdoc, NSF grant to study floral and faunal assemblages. And so I think if my understanding is correct, in 2008, Jack, Russ, Eric, Steve Jackson, many others who are here got a NSF grant to fund the database development of Neotoma. And then in 2009-ish, they got a grant to fund science that tried to use this new database that was being developed. And so that was Jack, Russ, Steve Jackson, and Eric. And so those were sort of the PIs on the grant that I was a postdoc on. And, and so I was a vertebrate paleontologist coming into a pollen lab. It was a whole new world for me. It really opened my eyes to the wonders of pollen. Lots of wonderful discussions with Eric and many others on the nuances of age models. And really just the power and also the frustration of databases. And so my sort of time with Neotoma has grown and sort of evolved since then. So I'm currently the associate chair of the database. When Russ retired, I took over as the lead vertebrate steward, and I'm also a member of the IT team. And so Simon.
Yep. And so I had been working during my PhD with data from the North American Pollen Database, trying to bring together data from British Columbia. And with my advisor, Rolf Matthews, I went to Bonn, Germany for the IPC meeting there. And that's where I first met Eric Grimm, talking about a climate transfer model that I had built that probably people shouldn't use. But I was finally introduced to Eric properly at a Neotoma meeting in Madison, Wisconsin in 2010, which I think was also coincidentally the first time that I met a young Earth creationist who was driving us on our school bus back and forth between the hotel and our meeting space at the University of Wisconsin. And so currently I'm the IT lead for Neotoma. I have been doing a lot of the work, doing the migration to Postgres, working with Socorro, building the new R package, and working on ways to try to upload like hundreds of records at a time, and really trying to figure out in a lot of ways what Eric had done and the reasons why things are the way they are. Oftentimes I'll think I'm smarter than Eric and then realize exactly why things are the way they are. So yeah. And so now we're going to talk about lessons we've learned. Is this still me?
You, Simon. Yes.
Okay, great. All right. You just cut in when it's not me. So there's sort of, we tried to lump this into four main things, is recognizing that people had are talking before us. And this really focuses on the sort of technical infrastructure of the database in a lot of ways. And so one of the things for sure is that details matter. And Eric had a very keen eye for detail, but also a very keen eye for what is important in the context of managing a large database. Context matters. And this is just making sure that if you are coming into the database, you fully understand the whole data ecosystem. Curation matters. And so making sure that the right people are the ones who are working with the data, making sure that data is checked along the way. And finally, flexibility matters. And Eric was amazing at building this data model that we can still use that now accommodates so many different types of records.
Okay, so to go in more depth on some of these lessons that we've learned, just the first one is about the details. I mean, I think this is a record stratigraphic diagram from the infamous Kettle Lake, which we've heard so much about in the earlier sessions in the Americas, focused on how amazing Eric was in the field, right? And he really honed in on a lot of the details. He assembled these amazingly complex, richly, densely sampled sites. He built beautiful age models. He had lots of, auxiliary data. And in the database, and then, when you're in the field, when you're collecting data, there's lots of data details that are really hard to capture when you boil it down into a database. And so those details, though, are what contributes richness to our data. And so I think the first lesson that we learned from Eric, and that he really built into Neotoma, is the ability to store and capture a lot of those details. There's 32 different notes columns in various different parts of the database. You can add notes about sites, about cores, about specimens, about age models, about really anything. Any data table, I think almost any data table has a corresponding notes column. And so this is just a collection of some of the notes that are associated. Some values are initially input as less than a specific value. And so capturing ways to provide context to the value. And then, the second part is that the context matters. It's not just about the values, it's about the types of values that are stored. So Neotoma doesn't store just an age. We also store the ability to document what kind of age it's generated. Is it a lead 210 date? Is it a C14 date? Is it a core top? Is it an estimate? We can document how data were collected, what it was collected from, what kinds of materials are being used to generate a date. And this is just, the metadata that goes into the date part of the database. And that metadata is rich and cascades throughout the entire database. And so, I think values stored in a database are only as useful as the metadata that are associated with them. And the infrastructure that Eric built in Neotoma, originally sort of cascading up from the original constituent databases, was just rich with context and rich with the ability to capture this amazing ecosystem of metadata. Simon.
Yeah, and then when we think about all of the kinds of information that we can put into Neotoma, it becomes really clear that curation is extremely important. And when you look at the software ecosystem that Eric built, to manage that data ingestion from just, the raw Tilia files that impose some sort of structure, the actual checks that happen in Tilia, for those of you who have done stewarding, where it checks, are there already sites that exist here? Are your lat longs reversed? Are they the right order? Do I find publications? These sort of incremental checks in the software, in the file formats, in the APIs, and then in the database itself are all built in there to ensure that, as much as we can, when data is in Neotoma, it is high quality data. And I think that's been one of the real big strengths of Neotoma, both as a community and as a service, is that it does give us this, we know that when data is in there, we can generally trust that data. Next slide, I think, unless, was I supposed to say something else? But it's not just the software. And one of the other amazing things that people have talked about with Eric is his ability to connect with people and to build a real community around the database. And so it's not just making sure that Tilia works, it's making sure that he has taken the time and is available to the stewards, the people who are working with Tilia, the early career researchers, the senior researchers who are trying to get their data into Neotoma because they've published a paper and haven't thought enough about getting their metadata done early. This fact that really, again, one of the strengths of Neotoma is the community that Eric has built around it. Next slide, I guess. Is this you or is this me? I can't remember.
This is you.
Okay, great, thanks. So part of the reason that it's so important to have all this curation and this community is that Neotoma, and I think one of the tributes to Eric is that because of this structure, because of this flexibility and the notes and all this kind of stuff, Neotoma is super complicated. There's 155 individual tables. Andrea, showed us that slide. The reality is actually much more complicated than that. There are 10 million total rows of data representing, I think Jack showed 7 million unique individual data elements across 40,000 sites. And next slide. To do this all and to power everything, Tilia, APIs, all this kind of stuff, it's 11,000 lines just of SQL. And so probably that's pretty abstract for a lot of people. You can think about how much R code you might have written. It's probably a lot. SQL is a very peculiar coding language, and I would never have, I mean, the fact that he's written that much, plus all the C code that he's written for Tilia, plus all of whatever else is just, it's mind boggling. It winds up being about 515 database functions to be able to do the stuff that we do on a day-to-day basis and come to expect from using things like Explorer. Next slide.
Still you.
And so ultimately, all of that really does serve the fact that Neotoma has been able to become a database of databases. It started out with pollen, geochronological records, and vertebrate fauna, but because of this very flexible data model, because of the work that he put into building the data model at a very early stage where everything is like Andrea mentioned, normalization. This is sort of a goal in a lot of database design. Because things are so highly normalized, it means we can add new relationships to the database as we bring in new and much more complex data types. And so it's easy enough to have pollen, and maybe we can say, yes, test data amoeba are pretty similar to pollen in terms of the general structure of their data. But as soon as we start getting into like specimen isotope geochemistry, biomarkers, these other things that require other supporting tables, as soon as we start adding in new different age types and age models, the database model has to change with it. And Eric has given us this incredible grounding foundation that lets us grow in a really amazing way.
Yeah, and so I think, when we were reflecting, these are all really important lessons that we've learned, just the sort of technical details of the database and the underlying infrastructure that supports Neotoma, that originally supported NAPD, that was built into Neotoma and really used to integrate databases from around the world. But I think, Eric's real legacy was, and what he's a great example of, is the power that comes with one person, right? And that power that you have at a very local level that then scales up to this global level. And, if you just look at, these are in orange, these are just sites across the US that had Eric's name associated with them in the publications in Neotoma, using Neotoma Explorer from last night. I think this vastly underrepresents his impact on particular sites, as we've heard in many ways earlier today. And it's been clear just through the science he's done at Tulane, at Kettle Lake, at Moon Lake, at Crystal, right, that he had a major contribution through the incredibly detailed individual records that he and his collaborators documented and built up and published on, right? And his record just as a palynologist alone and contributions to science through publications is amazing. But when you look at what he's built through the NAPD and through Neotoma, you can see that, you can take his sites, and through the NAPD first, you build and you integrate Eric's site and put them in context with all the other sites that are in the North American pollen database. So that's the snapshot here. And then today, where we're at now is, integrating those sites with all of the other pollen sites that are stored in Neotoma today around the world. What I'm not showing is all the other kinds of proxy, right? This is just searching on pollen. And you can see how Eric's contributions scale from single sites up to, his career of aggregating sites across space in the US, to North America, to the whole world. And, I think if any one of us had just an iota of that impact, we would call it a fruitful career. And to see this, writ large, I think really speaks to the power of small data in the long tail of data generation, and the power it has when you combine it in public open databases. Simon, you're muted.
Sorry, I was trying to be a good collaborator here. And I think that, again, just to bring this back to this idea of community, Eric has recognized that the success of his work rests on the fact that he was an amazing collaborator. He was a person that you could go and approach and talk with. And he really was able to place Neotoma at the center of this global community that, is remembering him in this way. The fact that we're doing this to remember Eric, I think, speaks volumes. And he built this. He built this thing that supports the people who are contributing their data, the people who are using data as educators, people who are using data from Neotoma to build new applications. And the lessons that we've learned from his work really help inform not just paleoecology, but open data. Concepts in open data are, people are learning from Neotoma, as Andrea as Andrea mentioned earlier. Ultimately, Neotoma is a data object. It is a database, but it is a community as well, and it brings people together from many different backgrounds, many different locations, many different interests, and again, that is the strength of Eric Grimm. I think, you know, go ahead. I think that's the end. And Jessica's going to talk now.
Sorry, I wasn't sure when you were ending that slide, but you know, I thought it was a good transition to the fact that Eric, you know, shows the power not just of data writ large, but the power of one person writ large through his impact on community, through his generosity of time, right? And so we've seen so many pictures today of different people. These are just two of the ones I have from a recent virtual event that he and I led with the Kaba folks in Australia, the Indo-Pacific, and there he is in the center of our virtual screen listening. And then this is a nice image from our gathering, the Neotoma community meeting in Minneapolis in 2019, right, where we're soliciting community feedback, trying to use sticky notes to generate community consensus about which ideas are the best ones to work in. And, you know, here's Eric up in the front, just like diligently examining, cataloging every single idea, and really just trying to think about all the different ways that you know, we can use the community to help improve Neotoma, you know, for the good of science. And so, you know, thank you.